Okay, well, good afternoon, everyone. For those that don't know me, I'm Paul Digard. I'm one of the co-directors of the UK-ICN. Um, so, bit of housekeeping. As most of you who have already joined will have noticed, the meeting's being recorded. So the talks will end up on our YouTube channel later on. Um, but also anything you say will be recorded as well. So by staying in the meeting, you're agreeing to that. Um, looks like everybody who's logged on is using their full names. That's good. Thank you very much. Um, Jill has asked me to keep an eye on the UK-ICN website, though we don't actually have anything specific to advertise right now, but that will change in the next month or so. Um, so with that, that's time to introduce our speakers. We've got a double act today from the Roslin Institute. Going to be talking to us about the novel feline coronavirus, and it's the team is Christine Tait Burkhard, who's a group leader at the Roslin, and Amanda Warr, who is a BBSRC Discovery Fellow, also in the institute. Great pleasure to have them here. I'm not going to give their biography. I forgot to check it in advance, and if I do it from memory, I'll get it wrong, and they'll laugh at me. They can tell you themselves if they wish to. But with that, Christine, I think you're kicking off the first bit of the talk, aren't you? So if you want to share your screen and get going. Or if everybody could remember to mute their microphones while our speakers are talking, please.
Brilliant. So hopefully everyone can hear me, but I'm at least unmuted. So let's start there, and I can see some of you still, so that's even better. So yeah, I'm Dr. Christine Tait Burkhard. As Paul said, I'm a group leader at the Roslin Institute, and I have been working on these things called coronaviruses that don't need introductions anymore these days for good grief, the past 14 years, something like that. So an old love of mine that has definitely come back for all of us to hate it. But this is a different story to SARS-CoV-2 that we're going to talk about today. And we're talking about what Kat dragged in today, the emergence and spread of a feline infectious peritonitis due to highly pathogenic canine feline recombinant coronavirus in Cyprus. So a little bit of background to FIPV to begin with. Feline coronaviruses actually sit in one big species family, for lack of a better word, of coronaviruses, the alpha coronavirus 1 species. And that encompasses, as you can see here, let me just get the pointer here, it encompasses canine viruses, feline viruses, and porcine viruses all in one species. And the rationale behind this grouping being that these viruses have a tendency to recombine a lot. And if you look at the evolutionary history, it's as complex as the different variants of the virus that you can find. This actually covers a fraction of the recombinants that you can actually find out in the field. But basically, we have two main serotypes of feline and canine coronaviruses that have originated probably in the canine coronavirus from an ancestry virus, but for the feline coronavirus, they have resulted through a recombination with a canine coronavirus to form serotype 2. But then there's lots of shades of that in dogs, but actually also in cats. And then we have the gradual evolution of a porcine coronavirus called transmissible gastroenteritis virus, which was quite economically important in the past. But luckily, this virus evolved, forming a partial spike gene deletion, and basically became its own natural vaccine. So the porcine respiratory coronavirus actually causes relatively mild respiratory symptoms, but pigs that have previously been infected with PRCV are actually protected from TGV infection, and there's a good maternal transmission to young piglets as well. But we're not here to talk about pigs today, we're actually here to talk about cats. And in cats, the story of feline coronaviruses becomes even more complex. Because in cats, feline coronaviruses are kind of both benign and pathogenic. There's two big different theories. One is the avirulent theory, saying that both a virulent strain as well as an avirulent strain are circulating. That's probably the less prevalent theory. The other one is the internal mutation theory, with various shades of whether feline genetics play a role, whether the exact starter virus plays a role. But let's go into what the internal mutation theory actually is, and then everyone can basically make up their own mind of what they think about it. So feline coronaviruses infecting a cat is usually orofecal, and the virus causes a relatively mild enteric or subclinical infection. Then the virus is called feline enteric coronavirus. It passes through the cat relatively easily, it sheds at high loads, but it infects primarily, not exclusively, but primarily epithelial cells in the intestinal tract. However, due to unknown reasons, the virus seems to mutate in the host, and then it changes its primary tropism and is much more prone to infecting monocytes and macrophages. It becomes systemic and then causes that systemic disease, including a cytokine response that exacerbates disease in many cases, as we kind of know from many coronaviruses, that being the case. So the host response is almost worse than the infection itself. However, it's thought that in this infectious form, in this symptomatic form, feline infectious peritonitis virus is much less likely to shed because it infects less of the enteric tract, and hence this virus is more or less contained to an individual cat, or at best an individual household where it can potentially infect further cats. Of course, other factors play into this, and we know that host genetics play into this, but what actually causes this mutation to occur, or whether there is indeed some sort of mixed population of viruses circulating, is still unexplored in detail. So to get to our topic for today, feline coronavirus in Cyprus, and what has happened. So colleagues at the vet school, specifically Harlampos Tipa, actually alerted us and Danielle Gilmore about an outbreak of feline infectious peritonitis in Cyprus that was going far beyond the usual cases that the island would see. So veterinarians on the island would see maybe 10 cases a year, maybe a little bit more, but nothing like the cases that they started seeing at the beginning of 2023. And based on the numbers that were circulated by shelters, an extrapolation of these numbers resulted in the headlines that feline coronavirus has killed 300,000 cats in Cyprus. This made the rounds in the UK newspapers, but of course this was also something that we were very interested in, together with our colleagues, to get to the bottom of what was actually happening in Cyprus. If we look at the Cyprus cat population, it is a lot. The island is supposed to have around 1.5 million cats, and most of them are stray cats or feral cats, so they're relatively inbred. Very important to know, but of course that means a lot of cats per square meter, so to speak. In 2023, the veterinarians reported to have treated about 10,000 cats with FIPV, but actually the stray shelters, they're not shelters in the common UK sense, they're mostly kind of places that feed and look after stray cats in bigger numbers, and they reported to have seen losses of between 30 and 40% of their cats. This one shelter here in Limassol say they've lost about 100 of their 250 cats that they used to feed and look after. So we may not be far off the 300,000 mark if you look at these numbers. So what happened? It is a bit unclear where the entire outbreak starts, but the initial epicenter was definitely Nicosia. Nicosia is obviously a divided province and a divided city, and we know that Turkey, or the Turkish part of the island, has had an increase in FIP cases, but whether it started in the Cypriot Republic part or in the Turkish part is unknown, but it's definitely where the epicenter started and from where the virus kind of spread throughout the island. I think I've got this right. Famagusta here at the tail end was definitely the last one to have the most cases. So there's clearly kind of a geographic spread throughout the island. And as we can see, we had one major big wave from January to roughly August. And the display here is only the PCR confirmed cases. But we also have another slight uptick here at the tail end. And what's interesting about this tail end is that it looks a little bit different in clinical presentation, and that it is tentatively more dry and more neurological form, but we'll get into that in a second. You probably wonder what's been happening since the beginning of the year. Well, December and January were quieter, but still above average from what was previously seen in 2020 to 2022. But interestingly, we see another uptick in cases now since roughly March, which could potentially also be linked with the birth of new kittens, of course. When we look at the kind of clinical features that we see in these cats, we also see quite a lot of differences in that the mean age is far higher than we normally see with feline infectious peritonitis. So the mean age is roughly 4.5. So this is no longer a disease of kittens on Cyprus, which indicates that pre-existing immunity probably doesn't help with this FIP infection. We also see far higher rates of neurological cases. So normally we see about 14% of cases presenting with neurological symptoms. There's two potential explanations. It's indeed a different tropism of this virus or better education of the veterinarians in recognizing neurological cases of FIP. And then lastly, what really startled us is when we looked at the first immunohistochemistry stainings of this Cypriot FIP infection. And when we normally see kind of primarily a villus tip infection in the colon here, very sparse IHC staining antigen detection, is actually very widespread in the colon of Cypriot FIP cats, and it looks a little bit like a different cell type being infected. And at this point, I'll hand over to Amanda, who's going to tell you all about what is actually happening with the sequences of these coronaviruses.
Okay, yeah, so bear with me because there might be a little bit of a lag because I'm controlling Christine's computer. Just gonna give up remote control and ask for it again, Christine, because it doesn't seem to be letting me do anything. Oh, there we go. Oh. No, 1 second. Get there eventually. There we go. Okay, so yeah, in my past research, I've done research designing tiled amplicon schemes to amplify and sequence African swine fever virus and porcine reproductive and respiratory syndrome virus. For those who aren't familiar with these schemes, this is kind of a picture of my African swine fever scheme. And the idea is we want to amplify the entire genome in small amplicons that are roughly the same size as each other and that overlap each other by a roughly similar amount. And what we then do with these amplicons is amplify them in 2 separate PCR reactions so that none of those amplicons within the PCR reaction overlap with each other. So we don't end up just amplifying these little short overlaps because we've mixed the wrong forward and reverse primers. So we then do, for every single sample, you're doing 2 different PCR reactions. And at the end, what you're hoping to get is reasonably equal coverage of all of the amplicons that cover the entire genome. And then there's a few pipelines that exist that we can use to put them back together again. So I was asked to design a scheme to amplify this new strain that's going around in Cyprus, to get a look at the genome and see if it could help us to understand anything about what was going on. And I said, sure, that shouldn't take long. Spoiler alert, that's still not currently working. It is almost working and should be working very soon, but it was not. This is not a quick process. This is a very fiddly process. So the tool that we usually use to design this sort of scheme is called Primal Scheme. So this is one that has been used. This was used for the SARS-CoV-2 primer schemes and also for Zika and Ebola before that. And again, the idea is to design these roughly equally sized amplicons with roughly equally sized overlaps. And it requires a multi-sequence alignment of whole genome sequences that are closely related to the outbreak strain. Of course, the problem here is that we did not have those. There are not many feline coronavirus sequences available in public databases. Most of the ones that are available are not feline coronavirus 2, and serology had told us that it was a type 2. So those are the ones that we really wanted to be able to see. And the ones that are available, they're very divergent and not necessarily particularly well annotated in the databases. So you've got viruses from all over the world from different years that cause different disease status. So we don't know if they're FIP or not. And of course, we've got these 2 different clades as well. So, you know, 85 genomes just isn't enough considering all the variables in there. So this ended up being a very manual process. We did use those multi-sequence alignments, and we did start off with some primal scheme designs, but it did end up just being us manually redesigning a lot of these primers and introducing degenerate bases to try and make them work as broadly as possible. So we went for 800 base pair amplicons with a 50 base pair overlap. And then the idea, once we've designed this scheme, is that we take our samples. These can be from a number of different sample types, including fusion fluid, blood samples, cerebrospinal fluid, and fecal samples or swabs. We get those samples and extract all of the RNA. We do a reverse transcription to get some cDNA, and then we go through our 2 separate amplification pools for the PCR. Once we've got those PCR amplicons, we then prepare those for sequencing using nanopore library prep. Up until now, we've been using the 112 chemistry, for those who are familiar with nanopore. We're just about to start switching to the most recent chemistry, 114, which should give us better throughput, which will be great for when we have this scheme perfected and we can just power through lots of samples. So then when we get out the other end, I have a pipeline called Lilo that was developed for working with African swine fever virus. So basically that is creating a consensus from all of the reads for each amplicon and then stitching them back together. And that's what that pipeline does. I won't go into too much detail on that, but the idea is to both get rid of any lingering nanopore errors, which are getting quite few and far between these days anyway, and also to try and stitch them back together to get a whole genome assembly. So this didn't work. This was the first attempt at doing this. And I apologize for how ugly this is to look at. But essentially, this is showing you along the top here, all of the amplicons that were in this scheme. And this is some of our samples. And where you see kind of this dark green, these amplified as expected and with very good coverage. The pale green ones had very low coverage, not enough to actually use for the assembly. The dark blue are the wrong size. So basically we've managed to skip out some primers in the middle and amplify a bigger amplicon than we had intended to. And some of these wasn't such a problem because they came out very high coverage, and I was like, well, I'll just use those then, that's fine. But some of them similarly were very low coverage. So this was a bit of a hit and miss endeavor. But there we go. One thing you'll notice is that this big white patch here is roughly where you would expect the spike to be. We really want to be able to see the spike, as that is quite often involved in this in-host mutation to cause FIP. So it was very disappointing to see this big blank spot right where we really wanted to see most. So we ended up designing an amplicon, a very long amplicon that goes from just the inside of amplicon 27 here to about amplicon 33 there. And that worked. So we managed to sequence one other comment I forgot to mention here is that we have one cat here that's slightly different than the others, subtly so, but this cat here has a very boring regular feline coronavirus 1 that is nothing to do with the outbreak. So there are still other strains going around in Cyprus, but the vast majority of them do not have that strain. So we managed to sequence our spike gene with a 3.7 kilobase amplicon, and of our 93 samples, 45 of those amplified successfully. The rest of them were just, there wasn't enough virus in there to amplify such a big amplicon, essentially. But yeah, so now we can actually see that amplicon. So on the next slide, I will be talking a little bit about 3 regions of the genome. So there's one here just to the left of the spike gene itself, and then a region just to the right of the spike gene, because these amplified so well in these cats, and we had good coverage now over the spike of more cats. So the first thing we noticed, or that I noticed when I looked at these samples, was that they were not feline coronavirus 2, which was essentially the only thing I thought I knew about this virus, is that it was going to be a feline coronavirus 2. What we're seeing over here, this is a tree of, basically I've taken all of the feline coronavirus and canine coronavirus genomes that I could find, and I've made a tree based on each of these regions. So the pink labeled samples here, these are feline coronavirus 1. The blue ones are canine coronaviruses, and the brownish ones are feline coronavirus 2. And the ones that are highlighted in yellow are cats from Cyprus. So it's very clearly sitting with the feline coronavirus 1s. It is not, as I was expecting, a feline coronavirus 2. And the other end of the genome also agrees with this, that it is a feline coronavirus 1 and not a feline coronavirus 2. But when we look at the spike, we see that it's actually not sitting with the feline coronavirus 1s or the feline coronavirus 2s. It is sitting comfortably within the canine coronaviruses. So this is kind of the moment where we realized that, you know, no wonder the amplicon scheme didn't work. We're not even trying to target the right virus. And, you know, we got quite excited at what was going on here. So let's move on to the next slide. So what's going on? We've already said that, you know, this species of virus, we've got a lot of recombinants going on. Does it really matter that we've found another feline coronavirus 1 to canine coronavirus 2 recombination? That's exactly how feline coronavirus 2 came about way back when. So the difference here is that the closest relative to that spike gene is actually this canine coronavirus called CB05. And CB05 is known as a pantropic canine coronavirus. So this has been going around in Europe for quite a while, and it's quite a concerning virulent strain of canine coronavirus 2. So this is a hypervirulent strain. It was first isolated in 2005 from some dead puppies, and outbreaks have since been reported in Italy, Greece, France, and Belgium. And there's also some other cases from other regions of Europe that are suspected to also be this CB05 or closely related, because we know that these other dogs died of coronavirus 2A after a disease similar to the previous cases. So experimental infection with this strain has been done, and basically every tissue they looked in, they found virus. So this is why they've called it this pantropic canine coronavirus. And importantly, it's also found in the intestines. We said earlier that FIP does not transmit well once it becomes symptomatic because there is less of it in the intestines. This virus, you find it everywhere in the dogs. So based on the parts of the genome that successfully amplified, I then went back and designed primers similar to that one that I did for the spike that would span the remaining missing regions. And then based on the sequences we could see from those overlapping regions, we've designed a new primer scheme. So we've got ambiguous bases in there because we're not certain that all the samples I was looking at in that process were actually the same feline coronavirus 23 and weren't just the circulating feline coronavirus 1 that was still in the population. So this new scheme, it's performing far better. Currently, I'm in the process of optimizing the primer concentrations to normalize the coverage across the genome so that we can get nice, accurate, and complete genomes. And this is something that comes up a lot with these primer schemes. They're great when they work, but it takes a while to get them working. Because amplicons, when you've got lots of different amplicons in one reaction, inevitably some of them perform better than others. You end up with, even though the primer pair works, some of them are just less efficient than others. And you end up with some that have very high coverage and they're just kind of hogging the sequencing capacity essentially. So ideally what we want from this scheme is for all of the amplicons to be present at about 2% of the total reads. That's not realistic, it's not actually going to happen, but we definitely just want to sort out some of the ones that are underperforming. I don't think this is actually the most recent version of the primer scheme. I think it's doing a little bit better than we see in this slide. But essentially, we just need to knock down the performance of the really overperforming ones and increase the performance of the underperforming ones, just to try and get it a little bit more even, just to ensure that this can be efficient and we can run lots of samples at the same time without worrying about dropout. So through this various redesigning of amplicons, I have got to a point now where we have a complete and assembled virus from the outbreak in Cyprus, but it is sort of a Frankenstein's monster of several different samples glued together at this point. And we are in the final stages of getting some assembled genomes from individual samples now. But what we're looking at here is a recombination analysis showing where this region that's recombined is. And again, this assembly, we've got a little gap here. This was the last remaining gap that we have since filled, but I have not had time to remake this figure yet. But we know that it basically, this recombination finishes just before the end of spike right here. So you can see quite clearly, we've got the green is feline coronavirus 1, and the purple is this pantropic canine coronavirus. And we can see very clearly that this region is the recombination region, and it's much tighter than the region was in feline coronavirus 2. And also you'll notice in this particular plot, so this is pairwise identity, you can see that the feline coronavirus 1 really isn't that similar. So this is the closest relative I could find for the feline coronavirus 1. And it's no wonder that the amplicon scheme did not work very well, because the closest relative in the databases is not that similar to the strain we're trying to sequence. But that's kind of a past problem now, because we now have some sequences from those cats and we can use those to design our primers. So the other thing to be aware of here is in fact, most of our samples have a deletion in the spike gene. And Christine's going to go into much more detail on what that deletion is and why it's interesting in a few moments. But just a note that this is very annoying when you're trying to design a primer scheme, because not only do we have this recombination, but we also have a deletion in most of our cases. And this complicates this tiled amplicon method, because some of our primers unavoidably sit in that deletion. So we're currently developing a second version of this tiled scheme that will perform properly on what we're calling the short spike, and will produce a similarly sized amplicon that spans that breakpoint where the deletion is, so that it will still work even if it's a short spike versus a long spike. So do we need to worry about this spreading outside of Cyprus? So anecdotally, there's been increases in FIP cases in neighboring countries and several European countries. And in fact, we've had a few fairly confirmed cases. Again, Christine will touch on that in a few minutes. Unfortunately, in Cyprus, given that there are so many stray animals there, we have these freedom flights that occur where cats and dogs are sent out of Cyprus on a weekly basis, primarily to the UK and Germany, for people to adopt them. And there isn't really any process for making sure that they are not carrying anything. So the chances that it's not going to get out of Cyprus are fairly low. And in fact, we've had a few cases that we've had to test so far. So this was the first one that came up. So this was a cat that was imported from Cyprus, presented with FIP. We knew that it was transported in a multi-cat flight and moved through 2 different multi-cat settings before it got to its home. And its home was also a multi-cat household, and we didn't know if it had outdoor access. Luckily, that cat was carrying a different feline coronavirus, so it may have picked it up in one of the households that it moved through, or it may have been just something that was circulating in Cyprus separately from the outbreak strain. But you can see the outbreak strain is up here, and this one is quite far removed from it. Unfortunately, not long after that, we had a second case where we were contacted by a vet who'd diagnosed FIP in a cat imported from Cyprus. Again, transported in a multi-cat flight, resides in a 2-cat household, thankfully no outdoor access, and a very cooperative owner who is very helpful in providing us with samples and is continuing to provide us with samples now after successful treatment, so that we can make sure that it's not coming back and that the cat is still fine. But this was very much sitting amongst our outbreak strain samples. So it has definitely arrived in the UK. It's unlikely it's gotten any further from this cat, other than the potential for it to have gotten into any of the other cats that were on the flight. But yes, she is now fine. She has now been treated and she's no longer carrying the virus. So I will pass back to Christine to give you some more interesting details on the spike.
Brilliant, thank you. So if we go back to the internal mutation theory, there's quite a bit known about the different genes that influence the potential change from feline enteric coronavirus to feline infectious peritonitis coronavirus. However, first caveat, much more is known about that switch in FIPB1, and it's mostly in the spike gene. So what we can look at is the spike gene of this canine coronavirus and see whether we have any amino acid changes that are reminiscent of this FECB, FIPB switch. And the simple answer of this complicated figure of all kind of amino acid changes that we have is that there's nothing obvious in simple amino acid changes. So the question is then, what is actually different about this virus that could make it an FIPB? First of all, the S1, S2 furin cleavage site that is present in some coronavirus is not present in FIPB2, and it's not present in this canine coronavirus. It's also got quite a, let's call it, bulk standard S2 prime site, which allows the fusion peptide to be released to insert into a target membrane. So again, there's nothing unusual about this spike that screams out. If we look at the receptor binding domain, and I have I have modelled the kind of mutations that we have onto this canine coronavirus that was found in some humans in 2018. And the group of David Visa have made brilliant structures of this canine coronavirus, and here modelled with the feline aminopeptidase N, which is the receptor for CCOV-HUPN. And as you can see, the kind of interaction is up here at this top loop, and that is where we actually have one change that could influence this, but the top loop that nicely inserts into APN, as well as the other top loop, actually have no amino acid changes of note. And David has actually looked at this structure of this FCOV23, and indeed this virus actually seems to be binding to aminopeptidase N, like most FIPV2s, and actually have a very similar binding structure to the canine coronavirus HUPN. So there's some indication that there's good receptor binding in this virus, and of course APN is very broadly distributed throughout different cell types across the body. So if that is the barrier to entry, it could indeed be that this virus has gained a broader tropism than other feline infectious peritonitis viruses that we've seen. However, what is obviously the biggest change is this deletion here to domain 0. And it's very reminiscent because we have seen it before in the alpha coronavirus 1 species. This is the deletion that happens between transmissible gastroenteritis virus and porcine respiratory coronavirus. And in transmissible gastroenteritis virus, this region is thought to bind to glycosides, so Nu5AC and Nu5GC. In other viruses of the same family, it's definitely sialicide binding. And in the general thought is that the use of sugar co-receptors is linked to the enteric tropism of these coronaviruses. However, in FCOV23, we see fabulous infection of the colon, so it doesn't seem like this deletion actually impacts the enteric infection at all. Interestingly, this deletion has been found in the field in other coronaviruses as well. So porcine epidemic diarrhea virus also shows this deletion. But again, in this enteric porcine coronavirus, we see an attenuation both in vitro as well as in vitro. However, there could be advantages to losing this domain 0. So this domain 0 is that kind of little dangly bit on the outside here. And it kind of makes the whole spike a little bit more rigid. If you're already not splitting your S1, S2, you're actually getting very rigid if you're that bulky at the top. And again, I've taken a picture from David's paper here. If you look at where the receptor binding domain sits, which is domain B, if you have a very rigid arm here, domain B becomes very difficult to access. But once you lose that, you get better access to the receptor binding domain. Of course, if you lose sialicide binding, it can be advantageous in terms of release from the cell surface because you don't get tangled back by sialicides. But on the other hand, it could impair your binding to the cell surface. But if you have stronger binding to your receptor, you might actually make up for that. So structurally, this domain 0 deletion could be very advantageous. But if we look at the prevalence of short spikes throughout the outbreak, we actually don't see any change in domain 0 prevalence. And actually in the latest kind of measurements were between the 50 and actually here about 70% mark. So short spikes still predominant, but it's not actually increasing and becoming the predominant version. So we still don't know whether they're both equally good at spreading or why short spike is there much more prevalent. Also in the presentation form, there's actually no difference between the two versions of the FCOV23. So stick with us to see what's happening. But I can share some preliminary data that my colleague Gary Whittaker has already shared last week, which shows that the short version of spike is very fusogenic. if it's expressed in cells that have APN receptor, it actually fuses to cells within about 4 hours of expression, which is nearly unheard of, and not a small syncytia, but a very, very big syncytia. So this could have very strong impacts on pathogenicity of the short version of spike. So the onset of disease could be much more rapid potentially than in the long version of spike. So let's get to summary. And what we know about this virus so far is that it's fast spreading. It seems to be more pathogenic than normal FIP and potentially the disease onset. It doesn't seem to be discriminatory of age or rather potentially immune status. And it is a clear recombination between FIPV and K9 coronavirus. And it's a beautiful recombination that's very reminiscent of SARS-CoV-2 and MERS coronavirus, where we see a beautiful spike recombination. So it's maintained all the accessory proteins that are feline adapted, but it has gained a new key to enter host cells. The highly similar sequences indicate that there's potentially cat-to-cat transmission, so horizontal transmission, which we really don't see very much in FIP cases. And there's a lot of virus in the colon. We see an increase in neurological cases and dry cases, which brings us to the conclusion that this is potentially a directly transmitting virus that has expanded its host range. What we're doing in the next steps, we are investigating more UK cases and sadly also small clusters. We've also got a reasonably confirmed case, 2 cases from Bulgaria, where there's actually a big trade in dogs or movement of dogs between Bulgaria and Cyprus going on. So there could potentially be an indication of carrier. Fast and accurate PCR diagnostics, virus isolation, and with collaborators looking at receptors, spike structure, and then continuing the epidemiology, the pathology, the vaccinology, immunology in Cyprus. But just a couple of slides of what we're doing with the diagnostics. So the diagnostics are actually sitting at this spike-spike recombination, which is very unique compared to both K9 coronavirus, feline coronavirus 2, and feline coronavirus 1. So it's quite unique to identify FCOV23, and this looks very promising. And the other thing that we're doing right now, and this is really data from today, so don't read too much into it, but it looks like we can isolate this virus on AKD cells, which is very similar to how FIPV1 behaves. We start to see syncytia-like formations after 8 hours, which is quite early in infection, and we see quite widespread syncytia at 24 hours. The infectious doses or the infectious particles numbers in the clinical samples don't seem vastly high, which could be sample storage or just that in effusion fluid there's not that much infectious virus. But again, lots of confirmation to do, so take this with a pinch of salt. We also see quite different types of syncytia, which could indicate the different types of spike being represented, but that obviously needs to be confirmed by PCR, by qPCR, how this virus grows, and potentially ideally with electron microscopy as well. So on that note, or on a more serious note, so will this spread to other countries? We don't know. It is very likely that it can be introduced into other countries, and it has been introduced in the UK. Whether it would cause equally devastating outbreaks, as in Cyprus, is very tricky to say because cat density in other countries is very different. There could be genetics at play. The cats in Cyprus are quite inbred. And we don't know anything about physical or animal carriers or virus stability. And one of the options that we have to consider is that we might not look at orofecal. If this virus has changed tropism, it could be oronasal transmission. It could equally be in ocular fluid. So cats love to headbutt. So transmission routes through the eyes are an alternative option of transmission. So lots of things to look into. with this virus that could slightly rewrite the story of how we look at feline infectious peritonitis globally. So lots of people to thank. You have heard from Amanda, who's done a lot of the sequencing work, but also Sarah, Marie and Andrew, who help with a lot of the virus work, DNA extraction, amplification. Lots of people in Cyprus to thank, particularly Dimitris and Maria, Harlampos and Stella here at the University of Edinburgh. And we have fantastic collaborators that help us to start to untangle the virology, which in particular is Gary and Annette. David helps us with structure to get together with Alan, Cecily, and with Volker, we're working on an infectious clone for this virus. And of course, we would not do it without our kitty cats, Amanda's lot and my lot. And we'd like to thank the entire team and hand over to you guys for questions.
Okay, well, thank you very much, Christine and Amanda. Open for questions. So there's one in the chat already from Jonathan Eubank. Does FCOV23 infect dogs? Which was something that I was going to ask as well.
Very, very important question. The bottom line is we don't know. We haven't seen any symptomatic cases in Cyprus. We have asked the vets whether they see anything in puppies, anything increasing. However, if you look at the cellular binding, the virus definitely binds to canine APN. So it's likely to be able to at least somewhat replicate in a canine host, which means it could be canine transmitted. But we have to remember that the pantropic coronaviruses have been discovered in Greece in 2005. So they have been around in that region for nearly 20 years. So there could be pre-existing immunity in dogs. So they could actually be properly asymptomatic carriers for this type of virus. And that's potentially also why we see the recombination in that region, that we might have co-infections. Whether that happened in a dog or a cat, my bet is as good as yours.
Does it infect anything else? Well, do that family of related species, do they infect any wild animals?
There's a very related rabbit coronavirus, but we only have a partial sequence. But other than porcine, canine, and feline, nobody's actually looked, but we're definitely wanting to see how broad even just the binding to APN is. We know, good news, that it's not binding to human APN. So that's a first good news, that humans are probably not carrier. Well, they might be physical carriers, but at least not an intermediate host.
Okay,
so we've got
Janet Daly. So Christine, if you're going to answer questions as they appear, can you read them out just in case people aren't?
Yeah, so Kim had her hand up in the.
Sorry, I can't see that.
So I was just going to start with Kim.
Right, apologies. Yeah, I'm not seeing the reactions at all.
Yeah. So we'll start with Kim, sorry.
Okay, thanks. Thank you so much for the really, really nice presentation. I just was wondering, because you showed also in the histology that the colon definitely has some virus in there. How often do you see that there is feline coronavirus 23 in the feces? Do you know that already? Have you checked that?
All the feces samples we had so far were positive. So
Oh,
it
wow.
seems like it's basically always in the feces. We've not checked for infectious virus yet, but the RNA levels are reasonably high to indicate that there must be infectious virus in there. So I'd reckon it definitely gets shed through the feces. How it eventually infects is a very good question.
Okay, Janet Daly's put a question in the chat. Are short and full length spike found separately in cats, or do you ever get mixed infections?
So far, we've only seen one or the other. It wouldn't necessarily surprise me to find both, but so far we've only found one or the other.
Yeah, we've actually run PCRs across all of our samples to just see what is short, what is long. None of them are mixed. So very strange, because yeah, it is a question. We need to start looking into different sample types from the same cats to see whether it's, because at the moment we have effusion fluid, cerebrospinal fluid. But again, I looked at whether the sample type is correlated with short or long. It's not. Geographic region is not linked to short or long. Presentation is not linked to short or long. It very much seems like they're coexisting, but still lots and lots of questions to figure out about why there's short, why there's long, and why one of them is not better than the other. Because from the in vitro infections, the short looks like it has an advantage, but obviously we don't know how that then translates to the physiology of the cat.
Okay, another question from Lorraine. Can you differentiate the viruses serologically?
No, not yet. It's a serotype 2, but we have a potential region that would potentially lend itself to actually design novel antibodies that could specifically identify FCOV23. So we're looking into that to indeed actually start some serology. Interestingly, based on the epidemiology, I would reckon that most of the cats will have antibodies now against this new virus. And what we're seeing now is kind of potentially the resurgence of susceptible individuals through birth or through loss of immunity. But this is something we really need to monitor over the next 2 to 3 years to see how this looks like over time.
Any more questions? So I cannot see if people have got hands up or not. So if you're sat there waving furiously, please just speak up.
Hello, it's Jin Chow here from Nottingham. Is there any evidence of airborne transmission of FIPV virus?
There's nothing evident. I mean, there were slightly bigger outbreaks reported in Japan, but again, they were thought to be orophycal transmissions. In the households, of course, cats are often sharing cat litter trays, and that's kind of where we've seen FIPV clusters, if anything. But it's almost an experimental question that should be tested, but of course it's very, very difficult to purposefully infect a cat with FIPV. Ethically, it's quite a tricky thing to do, even though obviously it could be infected and treated, but not an easy experiment to design. So the only thing we can do is look at, and that's what we're actually doing in the epidemiology, is look at oronasal swabs, look at eye swabs, and look at fecal swabs to see where there's virus, and equally looking at the lung epithelial cells, whether they're infectable, whether there's basically guilt by proxy, to figure out whether that's a potential infection route.
Okay. Well, we've got time for one more question, one or two more questions, if anybody has one. I have one for Amanda, actually. Amanda, if Christine manages to isolate the virus, then will you be able to use nanopore technology to directly sequence the RNA?
Well, the plan is at the moment to try and do it on the cDNA without the amplification. If we can produce as much virus as we want, that gets a lot easier. But the direct RNA, technically, yes, but the accuracy is still not good for nanopore RNA sequencing. They've spent so much time trying to improve the DNA sequencing quality that they've neglected the RNA a bit, but I believe they are coming back to it. So we could do it. Whether or not it's worth doing is another question until they've improved the kits.
Okay. Okay, well, if there's no more questions, please can everybody just join me in thanking Amanda and Christine again for a great double act, or a really interesting new outbreak that hasn't turned the world upside down yet. Thank you very much, and see you all next month for the next coronavirus seminar. Bye all.